to recap the plagiarism stuff from last week, which would have been Friday, but I want to talk about it now. So if you have it, go ahead and pull it out. We'll just briefly go through these 5 examples. Then we'll switch over to our discussion of the Hendry methods and results. And then for the second half of class, we'll start on a new topic. Uh-huh. Oh, okay, excellent. I have my Sharpie. Okay, so for those of you who brought it with you, oh, okay. For those of you who have it with you, I wonder if anyone's willing to share what they put for what kind of plagiarism was shown in example 1. Some of these are quite obvious, some of them are a little bit less obvious. There's more than one that you might have put, so we'll have a discussion about that. I saw a hand here first, yeah?
I feel like there's a bit missing footnote in example 1.
So missing footnote or missing citation. There's several quotes throughout here, but only the first one has a citation associated with it in that sentence. If you're using a quote, you should put the citation for each quote, and it's common, although I don't know how closely we've enforced this because we usually discourage quotations in this course anyway. If you put in a quote and you've got the author and the year, it's a good idea also to put the page number so it's easier to find the quote. So a complete citation with each quote would include the author and the year. Anybody put anything else there? Yeah.
I put the resourceful cider because there's only 10 words on here that are not in the quotes that are actually their words, and all the rest of the words are straight from the paper.
Good. Yeah, so that's the other one that I was looking for, that almost all the content here is quotations. There's no reframing it in your own voice or in the author's own voice. Somebody else, what about example number 2?
Before we leave
Oh, yeah.
1, my way of framing example 1 is I wouldn't give you a 0 on the assignment.
Oh, yeah.
Thank you. I would give you an F on the assignment for really crappy writing.
Yeah.
So, yeah, because there's just nothing of you in there anywhere.
Right. Thanks for pointing that out. What about number 2, somebody else? And if you're not sure which exact title or type it gets, if you can see what the issue is, just share that. Did any of you try comparing word for word something that you saw in an example with the paper? Sure, yeah.
I said this one was too perfect paraphrase because it seems just extremely similar to the actual words that was in the paper. They didn't even try to make it into easy words.
Yeah, I'd go a little further than that, especially the second to last line where it says to change squamosal and parietal horn lengths a full standard deviation in 21 to 36 generations. That whole portion of the sentence is just word for word. So I think that I would agree with you that maybe the perfect crime kind of thing, but it's borderline photocopy on a lot of these sentences, and you can rephrase that. You can break it down into 2 sentences. You can explain what standard deviation in 26 to 36 generations means to a layperson. These are things that can be rephrased and reframed. What about number 3? Any sense of how you would change this, or what you think the issue might be if you were the grader and you had to make a decision here? One thing I noticed about it is that in the process of paraphrasing it quite closely to the original work, the student ends up with a run-on sentence anyway, or a sentence that's way too long. So breaking things up into clear, concise sentences in your own words can also help you avoid the too perfect paraphrase. Would you agree too perfect paraphrase on that third one? Do you remember?
This is example 3.
Yeah.
I wrote that from memory.
Yeah, okay. So,
Yeah, I didn't look at the paper at all when I read that. And I do tend to both talk and write in longer sentences. I don't know if you guys have noticed that. But yeah, I didn't look at any of that.
but ended up being very close to what's in the text.
Probably because I've read this paper, like, how many years has it been since 2004?
We're coming up on
That is a
2024.
difficulty. So there's difficulty in avoiding plagiarism for all sorts of reasons. One of which could be that you're just too close to the material that you're writing about. So when we give you the four preconditions of natural selection, the first one of those that I always write is I'm not sure, but I'm pretty sure that's pretty darn close to an exact sentence from the words of species. Why? I don't know. Stuff just gets stuck in my head sometimes. Also, the order in which we cover these, we go morals, being reproduced and can survive, traits are variable, traits are heritable, that variation traits lead to differential survival and reproduction. That's once again the exact order in which Darwin took each of those things in each of the chapters at the beginning of the origin of species. So there does come a point where you're just so close to the material that you work with, it's difficult to escape the original work somewhat, which is why you cite it ultimately, because even though we kind of copy that order, we do retrace things in our own words, but we're so close to the source material that it's difficult to kind of separate us from it in the same way that being told to eat greatly reduces future fitness. That's an exact quote from a paper that I just really like when I was in graduate school. So as you know more and more, it's harder to know what you didn't know before you knew it. And so what I generally do when I write a paper is as I'm writing, I will write along, and then I'll leave myself little place markers because I know that I didn't know that back in the day, whatever that was that I just wrote. And then I'll write another sentence, I'll leave myself a place marker, and then another sentence, and leave myself a place marker, another sentence, and leave myself a place marker. These open parentheses tell me there's some information there that I didn't know in the past. And after I'm done kind of composing my thoughts and getting order right, et cetera, it's my job as a writer to then go back and make sure that I'm giving credit to the people who said these things before I did. Because virtually everything in your introduction is probably something that someone said before you did. And so I give myself these markers because it becomes difficult to separate what's your now walking around knowledge because you've learned a lot about biology. And you're going to find this to be harder and harder as you get older. Now it's really easy. Everything is new to you because a lot of you kind of had these topics in high school like this. But as you get further along, it's going to become harder and harder to remember what you didn't know.
This approach is going to be especially important and potentially useful for you, although everyone finds their own way to write. Especially useful for you when you're working on your lab reports, where you might be citing multiple papers. Here, we're not super concerned about you looking up all the references that Hendry et al. Looked up to write their introduction, although you might include one or two if you feel like it helps you convey what you want to convey.
Notice also in example 3 that there's a citation at the beginning of that, but there's a couple of sentences there. As long as it's clear that you're continuing to deal with that same piece of literature, you don't have to put a citation in every exact sentence if it's clear from the writing that you're continuing to write But once again, seldom will you spend more than 1 or 2 sentences on an article when you're, for example, writing an introduction for a paper. And that's why we're slowly forcing you to condense down to the point that you're writing about these things in smaller and smaller chunks.
Yeah. How about example number 4? Anyone have a sense of what the issue is with that one? Yeah, go for it. Sure. Well, they did cite the so they did cite the young adult stuff, but I agree with you also that some of this other stuff reads like it came from somewhere, right? Like, did you guys know that before you came here that some predators leave behind signs of predation that can be used to measure natural selection? I mean, you might have known they left behind remains of their prey, right? But this idea that you can use that to understand natural selection maybe is something that you learned in the process of doing this. And I think there's another one further down.
You've got the full standard deviation thing in there again. That's not language that I would use or that we would expect you to use.
Right. So the full standard deviation, right? You guys should know what we mean when we talk about standard deviation, so that you can put it in the kinds of words that would be understandable to someone finishing high school who hasn't taken this class yet. Yeah, that's a good point. So jargon, the standard deviation, you could think of it as jargon to someone who's a level or two behind you guys now.
Jargon is tricky.
We talked about this a little bit in the video, but go ahead, your perspective on it.
Yeah, jargon is tricky because it might mask that you don't understand what you're talking about. And you can sometimes use it effectively and not know that you're using it correctly. And usually when jargon jumps out at us, it's when you've used a jargony sort of term and it's used inappropriately in a sentence and we're like, oh yeah, you don't really understand what that term is. So the trick is just to try and avoid jargon, rephrase jargony sorts of terms in plainer language.
Yeah.
And then we get 2 things out of that. Usually the writing is better, and also we get a sense that you understand what you've been reading in a better way than if you're just using a lot of jargon.
So if you feel like you've learned some vocab here and you know what some of these jargon terms are, ask yourself, would my folks understand that? Would my sibling understand it? Would my friend at another college who's also a freshman in biology but not doing this, would they understand it?
In your writing guide, there's one of the things that it tells you to do periodically, which is to define terms in the course of writing. So read that section, and if you do learn some jargon and you want to continue using that jargon term, the first time you introduce it, you might introduce it by explaining what it is, and then from that point on, you can continue to use that term. And not only you'll know how to use it appropriately, but we'll also understand that you understand what that term means.
That's a good point and should remind you of what I suggested. If you choose to use a quotation, after the quotation, you can explain what the quote means. Then I would know that you didn't just figure out the answer is definitely in this sentence somewhere, so I'm just going to quote that, right?
Once again, that chapter 6 in that writing guide is really good. How you want really good advice.
How about the last one? Anyone want to jump on the easy one? Yeah. Right. Good. Nothing cited there. Okay, so we've got Caden. Oh, do you have a fresh Sharpie, Paul? We've got 3 participation points to give out. All right. So, excellent. Oh yeah.
The 5th commitment: incorporate definitions into your sentences. So it's on page
All right. And that's a good guide when you look at those things like running jumps. If you just look up editing and writing and stuff like that, there are certain ones that we see a lot of that don't necessarily come up on Google if you just look it up really quickly. Like running jumps, we see a lot of that. It's common. I remember doing it as I was getting started. I still catch myself sometimes in editing my own manuscripts. But there are certain ones, that's a good example of one that if you just look up, tips for writing on Google or something, that phrase might not come up or might not be explained very well. So use this book.
So you guys understand
Yeah.
what a running jump is? Yeah, go ahead. Somebody said something? Oh, yeah.
Oh.
It's a sentence that doesn't really add anything. Dr. Reynolds' thesis advisor refers to information-free sentences, which one refers to as running jumps. It's a sentence or a fraction of a sentence, or even a whole paragraph in some cases could be a running jump. And think of it like when you're doing the running long jump, the important part is the jump at the end, but you have to run up to it to get enough momentum to actually get to the business of jumping. And we usually employ them when we don't really know how to start something. And so we start down a path before we finally get to the really meat of the issue. And then when we finally get to the meat of the issue, we look at all that stuff that came before and we realize that it's not really adding anything.
They can be long and they can be short, and they also can be used in transitions. One that I see kind of spreading across, I don't know, students' work and the internet right now is starting a sentence with the phrase, "that being said," and then you say what you're going to say. The whole narrative means the exact same thing, you just remove "that being said."
Yeah, so when we do the presentation workshop with our seniors as they're getting ready to present, I have a hypothesis statement that's from an old student presentation, and if you delete the entire first half of the statement, it reads exactly the same as if it was there in terms of information content. So the thing to do is just cross through that and press delete.
Yeah. All right, so now I'd like to take something like 15 or 20 minutes, I'd say, and have you break out into your groups and talk about the methods and results portion of the Henry et al. Paper. So what you want to do is once you all get into your groups, remind yourself what the big picture question is from last week. We've talked about it a few different times. Make sure you can say it and you all agree that you have consensus on what the big picture question is. And then go through these 1, 2, 3, 4, 5, and 6 questions. I didn't have them numbered here, but I wanted to blow them up in case you don't have them. But they are on Moodle below the Turnitin spot for this. And then at the end, when you guys are going, you can turn in what you've written in hard copy. And if you weren't here on Friday and you would like your feedback from the intro, so you can incorporate that in your final version for October 11th, just come by and see me or Dr. Kowinski, whichever one of us has these, and pick up yours. Only a few here. So get to it. Talk about these questions.
Well, I wrote this.
A while back. You want to edit anything?
No, I just saw that it said salient. That's a full word.
Oh yeah. Yeah, I just copied this from when you posted it last year.
Yeah, it's weird how you get to know your own writing. It's like, oh, that kind of sounds like me.
No.
They're measuring the
So hopefully this is fresh from last week. Hopefully this one is fresh from last week, and yeah, I'm gonna lurk. Actually, I'll just pass it down. Here you go on for later. Mhm. For later.
Thank you.
Yeah. For later. Well, just talk about them. You can talk about it. That was from last year when we were remote. Yeah, you're good. 50. So 12, 10. Yeah, 110. It's perfectly fine if you read the whole thing. It's just we want you to break out your drafts by section. Yeah.
Like, okay, I have a question.
Yeah, for
So
sure.
I'm confused on the salient feature. Like, I looked up the definition and wrote down the definition of stuff.
Oh yeah.
Could you better explain that?
Yeah, salient is like relevant and topical. So like the important point. Which question did I have that in again? In Oh yeah, okay. Discuss what the salient features. So okay, so there's a lot of stuff in the methods that you don't have to put in your summary because you've only got about a paragraph. For example, the model selection AIC stuff to figure out whether it was unimodal or multimodal. You don't need to know about AIC. That's something that will come much later in statistics. But understanding that they were using different methods or different techniques to assess how many modes there are, and that they use 2 or 3, you can listen if you want. That would be important. But yeah. hmm Okay, good. Yeah. Yeah, because half of all this is bringing you into the language of the discipline.
I would understand 3 different methods.
Keep that attitude as you progress. I'm gonna check. I had a hand over here. You guys okay? I'll come back around. Yeah.
We're just asking about the same.
Oh, important, relevant. So remember when they were using the linear plots to compare modality, the cumulative frequency distributions? Then they used to determine whether those cumulative frequency distributions actually showed that S curve that means bimodal or a straight line that means unimodal. They used something called AIC, a model selection criterion. You don't have to include that. All that AIC stuff is beyond what you want to put in the 1 paragraph summary. What matters is how did they assess the number of modes, how many ways did they try to tackle that, and why did they use more than one way to assess it, right? Like, the important thing is how do you assess how many modes there are, and they use multiple approaches. And then that's good, right? Maybe explain one of them. What's another thing that might be not so? They talk about where they got the data from previous years. All these other researchers that weren't them, like they got data from the previous published papers, or unpublished, office drawers where no one published what they recorded. You don't have to list all those people. Just say they made some measurements and they used measurements from previous researchers decades ago. That's what I mean by getting the important part out of it. Mhm. Yeah. So, did you all feel like you understood what we meant by salient features of the methods? Yeah. Okay. It's the relevant parts that need to be in your summary so that you can tell the story of what they did and why. So, for example, they were looking at what feature of the distributions of peak size, what kind of distribution. So those were the raw data that went into size, and then what were the 2 shapes of that size distribution?
Unimodal or bimodal?
Yeah. So their methods were all centered on determining how many modes are present in a given distribution, a given place at a given time. You don't need to talk about the AIC score for whether it's unimodal or bimodal in the cumulative frequency distribution plots. What you need to do is show that it's challenging to assess modality, and so they use these 3 methods, X, Y, and Z. Maybe focus on one of them. That's what matters, not the model selection algorithm.
Oh man, we don't have enough space for
I think I need to
Excellent. That's what this is for. To have you all look at it and talk about it for a moment so that you can feel like you can rewrite it. Right. Yeah. This is just for credit and participation in the process so you get a better score at the end in October.
October.
Yeah. That's when we put the intro, methods, results, and critical thinking all together. Any other questions while I'm here before I look at another group? All right. Hello. Just coming to see where you are and if you have any questions or anything. Yeah. Okay. What was your? Well, maybe you already talked about this, but what was your assessment of the space for time question? Have you talked about that yet?
We said it was a space for time experiment,
Mm-hmm.
and that's because it would usually be done um, at that specific. It's not redoable because it's going to be done at that specific time in the space. It changes over
Yeah.
time.
Good. Yeah. So they use these other spaces as like replicates, and they alter whether human population increase or not is the treatment. Yeah. Good. Okay. Cool. Did you all feel like you understood the word salient? Some people were like, "Oh, I wasn't quite sure what you meant by that."
I have no idea what that is.
It basically it means what are the relevant and important features, but in this context, what it means is what aspects of the methods do you need to make sure to include in your summary so that you can tell the story of what happened. And you don't need certain things like how they use the AIC to select the model or whether it was modal. You don't need that in one paragraph summary. Yeah. Yeah. Good. This. Yeah. Okay. Good. All right. I'm gonna check on them.
Okay.
Yep.
What do you guys exactly mean by space for time experiments?
Uh huh. So, what's the pattern they expect to see at Academy Bay?
What's the pattern of
Yeah, like what happened at Academy Bay, or what do they think happened at Academy Bay?
human impacts have changed the diversity of fish?
You want to expand on that or build on it?
Well, I was gonna say like they kind of like went from bimodal to like less, and then they kind of like came back up to be like they went from like bimodal to kind of like unimodal back to bimodal.
So at Academy Bay, they're still unimodal,
Yeah.
but your modality explains what he meant by diversity, which is good. So I'm glad that went together, right?
And like El
El Garapatero, yeah.
that's where they went still bimodal.
Okay.
Yeah,
Because that's the one that's not like popular.
right. So when were the beaks unimodal at Academy Bay? Unimodal. Sorry. Yeah, yeah.
Since it's 1960, it's
Right. That's when it was bimodal in the 60s, and now it's unimodal, right? So you had this one place, one time where there was a change from bimodal to unimodal. That's Academy Bay. We can't rewind and add humans again and see if the same thing happens a second time, right? Like, there could be other things that happened between the 60s and now, right? Like, the Matrix came out. Is the Matrix coming premiering the reason that the beaks went to unimodal? Then how do you know it was humans that caused the beaks to go unimodal, right? So if you can't replicate that and see if it's the same cause and effect every single time, you can substitute other places as replicates. So what has, go ahead. Oh yeah, they were talking about like if you increase, if you go from 5 people to 10 people, do the finches care? Maybe not. But if you get a certain population size, then they might start doing things to the environment that would affect the finches, right? So I think that's what they meant by threshold. So what didn't happen at El Grapatero?
what didn't happen.
Yeah.
They like stay the same.
Yeah, the finches stayed the same. And what happened? What didn't happen there that did happen at Academy Bay in terms of the environment?
There were like less humans.
Yeah, no humans.
And no
But they didn't. But did they have data from the 60s at El Zapatero? So that's how we're substituting a different space for the past at Academy Bay. You can think of El Zapatero as like the Academy Bay in the 60s because people haven't arrived yet.
But like so in here, I thought Academy Bay was bimodal, like based off these graphs.
Yeah, so in the 60s, it
And then
was.
it's like two is greater than one, so
In,
it's more bimodal.
yeah, so in 2004, what this means is that the model that they're using, this AIC thing, can't distinguish, but it has a slight favor towards two modes.
they explain it
And
that way
then
that it could be due to
Then the
something.
Barrera went from more unimodal to more bimodal, just
well, oh yes, this 75, right? So more or less unimodal, and then here more or less bimodal. Yeah, yeah, yeah. This part I think is the most confusing and
this helped me make sense of it.
really,
Yeah.
yeah.
Because I was getting kind of like lost in all the measurements to like see which one was
Mm-hmm.
which.
Yep.
And this was like actually like visual where I could go and see, oh, it was bimodal.
Yeah. Okay. Good. And it's got the nice little helpful um, lower part portions. Good. Okay. Let me check on time here.
Guess what? You ever bought birdseed? There's no scarcity of food causing them
Was that Ferrero Bay we were just looking at?
Yeah, there's
Oh yeah.
Gangrel.
Yeah, so Barrero Bay was the confusing one because it had narrow variation because there weren't any big beak finches. Yeah, but then you get more big beak finches later. Yeah. Yeah. What do we do? Right. Um, all right.
I mean, the point is you're substituting. You can't go back in time, so you're just going to substitute another place.
Good. What did you guys get for the primary results at the end? How'd you summarize that?
Said, but they took like all the different years and they looked at when bimodality like increased,
Mm-hmm.
and we said that they got that in the 1960s it was really strong, and then throughout from 1968 to like 1973 it decreased, and there's an increase in like medium size,
Mm-hmm.
and then in like the 2003 and 4 it started to increase again.
Yeah.
Maybe
Yeah. One thing you got in there, I was really pleased to hear, was the increase in the abundance of medium-sized finches. Right. That was one of the things that they found. They were able to say that because even though it went to unimodal, the variation was still the same. The only way to get that is to have more finches in the middle. Good. Okay. Cool. It sounds like, yeah, it sounds like we're doing good. Okay. Okay.
So it's food abundant. Why would you specialize on large seeds versus small seeds? If you're a shoe salesperson, you have a shoe store, and you carry loafers and dress shoes and tennis shoes, and then somebody comes next door and
I'm not sure.
I didn't take out.
All right. Anything else back here?
I think we get the general idea.
How did you guys summarize the primary results? Like, how short could you make your assessment of what they found in the end?
So what they found, I'm wrong, I think this is completely wrong, but basically what they found is that when you have the results and without the results, that the interpretations are the same.
Which results?
The
Wait, are you talking about the one where it's like that bimodality after 1960 the County Bay was weak and stable?
Yeah, I'm asking you basically what happened at Academy Bay and how is that the same or different than what happened at the other places?
Oh, well, one in Academy Bay was weak. The rest of them were like strong. Well, the other one was weak. That was because of the low population.
Yeah, you're talking about bimodality here?
Yeah. Is
Yeah.
that what we're summarizing?
Yep.
Yeah.
Yep. Basically, how did the distribution of beak sizes change at each place?
Oh, yeah. Academy Bay was weak.
Weekly bimodal when?
Yeah.
When?
What?
When? When was when were they? When was it more unimodal or less bimodal?
After 1960.
Yeah, recently,
Yeah.
up to the 2000s even,
And then,
70s to the 2000s.
but
El Grapatero,
yeah, that
Garapatero.
was strong. That is strong bimodality
When?
after 1960.
Right. And what's the difference between El Grapatero today and El
These
Grapatero today?
humans are in the Academy Bay.
Yeah.
But the
Yeah.
other one was also
Yeah.
Borrero Bay was
Yeah.
weak,
But they had a reason for why.
but because the population was low.
The population was low, and there were almost no large beaked finches there to begin with.
Yeah.
So at Academy Bay, you have a certain amount of variation, and it starts out as bimodal. Then you have the same amount of variation later, but then it's unimodal. So what does that mean about the population of finches in that time? Same variation,
Yeah.
but now we've added the middle finches.
Oh, they made it grew.
Yeah. Yeah. So the distribution goes the same, but all you've really done is add a bunch of intermediate finches that would have died in the 60s, but now they can survive.
So now when we write these like this page and a half, how much of we just got to go to paper to really cite like everything? We didn't know any of this.
Right.
So we got to go through.
No. So the advice he was giving is important for the literature, for the lab report, but here, within a single paragraph, you're only probably going to only be referencing the one paper.
Yeah.
So if you take something really closely, make sure you cite it. But if you have 2 or 3 sentences and they're all clearly flowing together, you don't have to cite it for every single sentence.
Okay. That's why, because
Yeah.
he was like, okay,
Right.
damn.
That comes for lab. Yeah. All right. I'm going to get us together. Yeah. All right. All right. So, nice. Okay. Let's come back together now. I want to talk about a few of these and then leave a little bit of time to get started on our next unit. So, let's go. So we talked to a few of you about what we meant by these salient features, right? What are the parts of the methods that are going to be important to get in your one paragraph summary of the methods? What are some of you come up with for that? What are some of the important things versus the things that you can probably leave out? Yeah.
It's important that you say what the experimental group was and what the control.
Yep. Good. That'll be really important to understand why what comparisons they're making and why. That's the idea with controls, is you can explain what comparisons are being made and why you would make them. Yeah. Right. And so what, if you don't need to include all the specific data, what patterns are important to describe? Mm-hmm. For sure. Yeah.
Yeah, like they talked about the data for Academy Bay,
Mm-hmm.
and like the specific n equals like 327. Like for an average person, that does not make any sense.
Right. Yeah. So these sample sizes for the different finches, you probably don't need to worry about all those details of how many finches they measured and which years. Speaking of which years and who measured the finches, who collected all these data that they used in all their histograms? Some of it's other researchers, and what do you say?
Like David Snow.
Yeah, so they give you all these citations or references to previous researchers who collected data. So it's important for you guys to know that they collected some data and they used data from previous researchers. But you don't have to reference all those people, right?
Jamie, I had to look up his birthday. Off the top of my head. On Jamie's introduction, he said that they collected all this data all the way back to the 60s, and like Henry wasn't even born until 1968. But I didn't know that. I had to look that up.
Yeah, good. And you guys answered that already a little bit. Okay, good. And what about this space for time thing? What do we mean by that? Yeah.
That's like if you can't go back in time, so sometimes I'll substitute another space.
Yeah.
Like the environment is like what it used to be back in time.
And why would it be, if you could go back in time and rewind the tape and run this again, why would you want to do that?
Because then you get what exactly happened.
Yeah, for sure. You'd be able to track each and in every single thing. But the other aspect that we talked about a little bit was, sure, the finch peak distribution changed and the population of humans increased. Are you sure that one caused the other? Right? Go ahead.
I'm probably completely off topic, or topic for that, but didn't they have to like go back and do a bunch of recalculation and kind of fix some of the data?
They did. So not everyone collected data in exactly the same way. And so in order to make the comparisons fair, you have to standardize your data or think about what kinds of comparisons you want to make. I think there was one group of researchers who didn't measure beak height, they only measured length or something like that. That's an example of where they had to adjust how they were comparing in order to account for that difference. Yeah. That's another thing that you could get a sentence or two in there about that, about the challenge, but really to tell the story of what happened in Academy Bay and why we think or why they think, the authors think, that it's the result of human influence on the environment, maybe you don't need that. Not incorrect if you include it, but it might not be critical to the story in 2 pages. So that brings us to this one, actually. So thank you, Lily. What did they measure? What were they trying to measure, and then what did they actually measure? This should remind you of last Friday, for those of you who were here. Anybody new? Anybody want to earn an extra point for their team today? All right, carry us through.
They actually measured, what they wanted to measure was size, but there's
Yeah.
like a lot of things that goes into size. So what they actually measured was the length, the width, and the depth, and then they did a principal component analysis, and they created the variable called size.
Yeah, good. Well put. So getting all these different dimensions in order to assess size variation in these histograms is what they were actually trying to get at. Good. We talked with most of you, I think, in our little groups about the different ways they analyze their data. What did you guys think of it? Which way of assessing the distribution did you all find was helpful, or more helpful or less helpful? How many of you liked the histograms? How many of you felt like you understood the cumulative frequency distributions better? How many of you just compared AIC scores? Right? Yeah, go ahead. I'm just kidding. Yeah, that's the cumulative frequency distribution, where you're comparing the accumulation of data, right, as you go from one side of the graph to the other, depending on whether it's bimodal or unimodal, you'll get different rates of accumulating those data. And that gives you those 2 lines to compare. Good. All right, and finally, someone tell me what happened at Academy Bay. Just generally, what do they argue happened at Academy Bay from the 60s to the 2000s? Someone new? What about from this group up front? Yeah.
It went from bimodal to unimodal.
Yeah, that's the argument they're making. That you had this bimodal distribution of beak sizes in the 60s towards a more unimodal distribution. Someone else, what happened to the total variation in finch beak size at that time? What about another way, the range of variation? Yeah.
I think we have an increase in like the intermediate size.
Good. So that's where I was going. So we didn't lose small beak finches and large beak finches, but they gained the intermediate beak finches, and that gives you that unimodal distribution. Okay. How did they relate that back to their original question of human impacts? So I don't know if they actually say that, but that's a reasonable inference given. So Caden said that finches were being fed by humans. I don't know if they actually say that, but the implication that human activities increased the food available for finches there, including food resources that the intermediate-sized beak finches could eat. So we went from scarce food resources to abundant food resources. Good. Anybody else have any questions about that or want to add? What about El Grapatero with that same point? What didn't happen to El Grapatero?
Unimodal distribution.
Yeah.
Stayed bimodal.
Yeah. Stayed bimodal, right? So you didn't have people present supplementing the food for the finches, and so the selection was relaxed, and they were able to eat all varieties of food, and lots of finches could survive and better pass their genes on to the next generation. Yeah. All right. So hopefully you all feel like these discussions you have with your group and this sort of class discussion at the end here helps you in revising your methods and results section and in putting this all together. Paul's giving you a little visual diagram here of what we're looking for on October 11th. Your introduction, which goes over some of those same questions that are in that How to Read a Scientific Paper document. What's the big picture? What's been done before? What hasn't? What did they try to solve and what exactly did they test? Those things go in about the first paragraph or the first half page of your final paper. Then you'll have a little bit on the methods, those salient features you've identified in your discussion today, results, and results will blend over into your conclusions likely here on the second page. We want the second half of your second page to be critical thought. Does anyone remember how we described critical thought in the context of this course a week or two ago? And remember what we're looking for when we say some critical thought. Racking them up.
You said don't say whether you think the paper is like good or bad, but you should say like what further research needs to be done or like how you would change the experiment.
Good. So follow-up experiments to better understand the story of what happened, or a follow-up experiment to test an assumption they made but didn't actually evaluate in this paper. Some other piece of the story. If you can pull together other pieces of the story that you might investigate, or a logical follow-up project that you would imagine doing if you had the money and you're going to Galapagos and you had a team and all that, what would you do? Good. I'll come around again with the pen in a minute. All right, so shake that off a little bit. We're going to change channels. I'm going to introduce to you guys this new unit, Inheritance of the Phenotype. This will bleed over likely into the next class period, and then I'll transition to Paul for the next portion.
Going back to the bird feeding thing.
Oh yeah, did they actually say it?
Yeah, it's on page 1892. Indeed, perturbation. So it's talking about. For example, humans might increase the relative abundance of intermediate-sized seeds by altering habitat, introducing non-native plants, and providing food for native and wild animals. Indeed, perturbations of this nature are prevalent in the Galapagos and are particularly obvious at Academy Bay. As one striking example, we are aware of at least two long-established finch feeders within 500 meters of our study site, these feeders can be readily cracked by birds having a wide range of beak sizes, albeit with different efficiencies. We mentioned this act of feeding of finches as just one example of how humans can materially change finch diets and presumably the adaptive landscape for beak size. So they actually go on to 3 or 4 sentences.
Yeah. So they've made observations, and they proposed a hypothesis.
They have.
There in the discussion section of the paper. Yeah. Which is something you guys might be doing as well in your discussions at the end of lab.
A good form of critical thought might be thinking, okay, they've put forth this hypothesis, how might I go about testing this?
Yeah. Okay, so one of the things we try to do here is structure some of the different topics around the 4 premises of evolution by natural selection. So today what we're going to do is the lead up to heritability, basically, the lead up to heritability. A while back, you guys talked about the central dogma. Can anyone tell me how you would explain what a gene is to somebody? What's a gene? Yeah, Emma.
Sequence of DNA, nucleotides, code for functional function.
Okay, yeah. So you read that one. That's a good one. I have something very similar to that up here. A section or sequence of DNA that codes for a specific functional product. In the context of this lecture, when I say functional product, I'm mostly going to be referring to a protein. A sequence of amino acids that's folded up into a large complex macromolecule that we call a protein. Proteins come in many forms. There are structural proteins like the keratin that makes up your hair. There are enzymes, proteins that catalyze chemical reactions. There's many different kinds of proteins, and our DNA codes for many of those. Current estimates are that there's something on the order of 20,000 genes in the human genome, although there's lots of different estimates there. And I think early estimates were focused mostly on the number of genes that code for proteins, but there are also many sequences that code for functional RNA molecules that then regulate other genes. So it becomes a really complex system, but you can think of a single gene as a sequence on a chromosome between that start codon that you had to find to set your reading frame until you get to the stop codon that says, okay, no more nucleotides, no more amino acids to translate. This is what defines the end protein product. Of course, genes are part of this double helix of DNA, which is folded many times over. And during cell division, we see DNA as these large condensed units that we call chromosomes. So we're going to talk more about chromosomes here in a minute. So I have another question for you. What is an allele? And I'm introducing you a term here. Genes occur at loci, at places on the genome, or positions on a chromosome. So it's a physical location, and if you talk about one gene at one locus, you use locus singular, and loci is plural. But this is the question that will appear down here. What is an allele, now that we've talked about what a gene is? So Caden says a trait that someone has, which is not exactly correct, because the trait that someone has that you can see is the phenotype, but it's related directly to this question of what is an allele. How about someone from the mitochondriacs? Mitochondriacs? Yeah. Yeah, a form or a version of a gene. So at a single spot on a chromosome, different people might have different versions of the same gene. There might be one of several genes that affect your eye color, right? We don't all have the same eye color, so that means we have different versions of these genes. So here's an example from a textbook, I think, where we have a single chromosome here. And on this chromosome, at this locus, there's a gene that is the version that confers purple color in this flower, this foxglove flower or something. Whereas on this chromosome, at the same locus, these chromosomes are homologous, meaning they have the same genes on them, but it's a different version of the gene at this locus here. So you got two alleles that are different, but the chromosomes themselves are the same or homologous. Now, you guys might already know this, but most organisms, most animals and plants, most animals and plants, I'll leave it at that, their mature bodies are diploid, meaning that for every cell in the body, there's 2 copies of each chromosome. So you have homologous pairs. So a typical person has 23 pairs of chromosomes, giving you a total of 46. Because you've got 2 copies of homologous chromosomes, we call that diploid. And it's often abbreviated as 2N if you don't know how many chromosomes that is. So any given typical animal that you might see, the squirrel, the hawk, the robin, you're looking at a diploid individual, but you probably have to look up how many pairs of chromosomes they have, right? I don't know how many pairs of chromosomes a squirrel has. But we use this 2N to describe a diploid, 2 times the base number of chromosomes. A cell, an individual cell, is called haploid if it has only 1 set of chromosomes. You can also have organisms that are made entirely of haploid cells. Most plants have a stage in their life where they're entirely haploid. Honeybees, right, have a haploid phase.
Yeah, the workers are.
The worker bees are haploid. So there are special scenarios where you might have a haploid organism, but for our purposes and for the stuff we're going to talk about when we talk about inheritance of the genotype, most things are going to be diploid, and that's going to be really important to understanding how cell division happens and how new organisms are built from their parents. So I want you to know the difference between diploid and haploid. Those are 2 terms you want to know. All right. So let's consider a cell for a moment. Most of you have when you've ever considered a cell before, you probably considered an animal cell. And because the genetics that we're going to learn about was originally worked out in peas, I thought I'd give you a plant cell. So plant cells, right, they have the cell wall, they got that big vacuole you've seen so many times, but the nucleus is where the action is that we're going to talk about. Typically, as an organism grows, the cells divide. Does anyone know the name of the process of cell division where you go from 1 cell to 2 identical daughter cells? Yeah, good. Everyone say it together. 1, 2, 3. Beautiful. Excellent. Love it. So in a plant, the way that this typically happens is you've got all this chromatin, all this DNA in a nucleus that's uncondensed. It's open. The molecule is available for enzymes to come in and read through and transcribe RNA, and interact. But when it's time for the cell to divide, it has to package everything up. Right? It's like you're moving. So the first thing that happens is all this DNA condenses into these chromosomes, and at the same time, the DNA is duplicated. So a single chromosome is often drawn like an X. That's what you get when the DNA has been duplicated. So a single chromosome composed of a single chromatid gets replicated, but it's still tied together in the middle by this thing called a centromere. And so this indicates a single chromosome that's been duplicated. So that's why it's an X, because it's been duplicated. Then these chromosomes that have condensed and duplicated align along the metaphase plate in the middle of the cell. They line up, and then those replicated chromatids are then separated and pulled towards either pole, giving you 2 new nuclei with the same number of chromosomes, except this time they haven't been duplicated because it's not time for this cell to divide yet. So this number of 4 of each, 1 of each color, or 4, is the same as this, except it hasn't been duplicated yet. And when you go through prophase, you duplicate the chromosomes to get those X's. I think it's a little bit confusing because we talk about whether they're drawn this way or this way. They're often both referred to chromosomes. But really, this is one chromatid, and when it's replicated, you get sister chromatids forming a chromosome. So I'm just spending a little bit of time on that because that's often a little bit confusing, but it doesn't have to be. So How many of you raise your hand? I guess I already guessed this. Are somewhat familiar with this? Do you feel like you understand something about cells replicating? Right? Okay, good. Here's what we're going to get into. What processes occur during sexual recombination? What kind of cell division is involved there? Right, meiosis. And then we're going to link this to how does this process of meiosis determine the patterns of inheritance of variable traits, the kinds of traits on which natural selection might act. So sexual recombination refers to, in the course of a life cycle, basically transitioning or alternating between meiosis and fertilization. A single life cycle consists of meiosis and fertilization and a whole bunch of fun stuff in between, like growth and development. So meiosis, reductive cell divisions, where the resulting cells have one copy of each homologous pair of chromosomes, or one chromosome each, one copy of each chromosome. And then fertilization is the union of two cells that are haploid to make a new diploid cell. So that's all it is, going back and forth from reducing, putting them together. In the process, we shuffle the genes up, and that's what gives us the patterns of inheritance that Mendel found. So here's mitosis again. We condense those chromosomes and replicate the DNA so you get the little X's, align them at the plate, and then divide to get two daughter cells. In meiosis, what's happening is not only are condensing the chromosomes and replicating the DNA, but also the homologous pairs pair up. When homologous pairs of replicated chromosomes pair up in the middle here, you get these things called tetrads. It's 4 chromatids all lined up together. And they're each still united by the centromeres based on where they've duplicated. Once they've lined up, and they do this randomly, this could have been drawn with the gray on top and red on bottom, or this could have been drawn with the blue on top and the yellow on bottom. Any combination, it's just they're pulled together and lined up. Like when we all asked you all to line up by height, we could have left it up to you which way you want to face, right? It doesn't have to be all facing the same way. It's just lining them up here in these tetrads. Then they get pulled apart. At this point, notice that each pole is receiving only one of each homologous pair. Is that clear? Each pole is receiving only one of each pair. Good, I see some nods. Good. If we then continue, so the thing about meiosis is you get this far, but each pole still has too much DNA because you still have the X's. So there's a second round of meiosis here, where then these are separated from each other. So you end up with 4 daughter cells, and each of them has only one chromatid, and only one of the 2 members of a homologous pair. So in the end, this nucleus over here will have 2 chromosomes, whereas the original parent had 4, right? Okay, so that's meiosis. I want you guys to be thinking about that. I've put a slide in here for you to look at that goes through the whole thing. And we're going to talk about Mendel's laws and his patterns that he observed and how they link to the actual biological process that he didn't even know about at the time. Oh, yes. I'm doing that for 5 points for people. Yeah. Yep. So I'll collect things here. Paul, can I have you hand